What if we could make a cancer diagnosis way more accurate and cut down on the need for follow-up surgeries just by changing how we look at things? Well, today we're going to dig into a huge shift in breast pathology that does exactly that.
Okay, let's just jump right into it with a question that unfortunately a lot of patients have to ask. Why do so many people need a second surgery after getting a tumor removed? It's just a huge source of stress and worry, and the reason why, well, it has to do with something you can't even see.
So here's the deal. When a surgeon takes out a cancerous tumor, the most important question, the thing that's on everyone's mind, is, did we get it all? They've got to remove that tumor with what's called a clear margin of healthy tissue all around it. The problem is, you can't see where a tumor actually ends and healthy tissue begins just by looking at it.
So why is finding those edges so tricky? Well, it really comes down to the old school way we've looked at tissue samples, which is basically in little tiny pieces.
This here shows you the standard process. A surgeon removes a piece of tissue, and then a pathologist, that's the specialist who examines it, takes these really small, thin samples to look at under a microscope. But the big problem, as you can probably guess, is that they're only seeing a tiny, tiny fraction of the whole thing.
And this slide, wow, this really shows you what the problem is. Think of the tissue the surgeon removed as a giant photograph. The old way of doing things is like cutting that photo up into a bunch of tiny squares and then only looking at a few of them. The pathologist is basically forced to guess what the entire picture looks like from just a few scattered puzzle pieces. In fact, get this, it takes, on average, 21 of these little conventional sections just to cover the same area as one of the new large format sections. 21! 21 separate, disconnected pieces to try and make one complete picture. You can imagine how easy it is to miss something really important in all those gaps. And this right here shows the fatal flaw of that old method. Those little rectangles in image A, that's where the samples are taken from. But then you look at images B and C, and you see where the cancer actually is. The samples can completely miss the edge of the tumor. That gives everyone a false sense of security, and that's what leads to needing another surgery.
So what if instead of giving pathologists a few random puzzle pieces, we could just hand them the entire map? Well, this is where a truly revolutionary new technique comes in and just completely changes the game. Let's walk through a case. In images A through F, you're seeing what the radiologist sees before the surgery: mammograms, MRIs, ultrasounds. These are the tools that help them figure out the rough location and size of the tumor. This is kind of like their initial battle plan. And then, here it is, the breakthrough. Look at image G. That is not a tiny fragment. It's a single large format histology slide. We're talking a microscopic view of the entire slice of tissue. What you get is a perfect map that lines up one-to-one with a radiologist's scans. For the first time ever, the whole team is looking at the exact same complete picture.
I mean, look at this. This slide just drives home how much better and more efficient this is. On the left, 45 small blocks. That's the old way. On the right, just two large format blocks covering the exact same area. The difference in how much information you get is it's staggering.
Okay, let's look at another patient. So those initial scans in A, B, and C, they're flagging some trouble spots. You've got these tiny calcium deposits called malignant calcifications and some distortion in the breast tissue. The team knows where the problem area is.
So after the surgery, the large format technique takes center stage. Images D and E are basically X-rays of the entire piece of tissue that was removed, and F is that amazing comprehensive histology slide. Now the pathologist can literally trace the cancer's exact path through the tissue, millimeter by millimeter. There's no guesswork.
And this is why it matters so much. See the arrows in image F? They are pointing to cancer cells that have reached the very edge of where the surgeon cut, the margin. With the old fragmented method, this is exactly the kind of thing you could easily miss. But here, with the full map laid out, there's no question. The team knows for sure what they need to do next. And here's just one more quick example to really nail it down. Again, that large slide in image F gives a totally definitive answer. The arrows clearly show the cancer goes right up to the surgical margin. There's no ambiguity, no guessing. This is what precision diagnostics looks like. Clear, actionable information. So what's the real-world impact of all this? Of going from tiny fragments to a full map? Well, it's absolutely profound. And it all boils down to two very simple things: fewer surgeries and better outcomes for patients. For the patient, this changes everything. It means a huge drop in the need for those stressful repeat surgeries. It means the diagnosis itself is way more accurate and complete. It also helps the entire medical team, the surgeon, the radiologist, the pathologist, communicate with perfect clarity. And all of that leads to way more confidence in the treatment plan and, at the end of the day, to better long-term health.
Now, a massive shift like this, it doesn't just happen overnight. It's really the product of decades of incredibly dedicated research.
The source material for what we've been talking about today, and the technique itself, comes from the absolutely pioneering work of Professor Laszlo Tabar and his international research group. Their vision was to finally bridge that gap between what the radiologist sees on a scan and what the pathologist sees under a microscope, creating one cohesive, accurate process. And you know, all this brilliant work really leaves us with a final, fascinating thought. This whole revolution in breast cancer diagnosis was achieved just by moving from a fragmented view to a complete one. It really makes you wonder, what other medical mysteries could we solve in all kinds of different fields if we just found a way to see the whole picture?